pleased to introduce our next speaker, who is Dr. Maria Hayes. So Dr. Maria is a senior scientific officer at the Tigarsk Food Research Centre, Dublin, and an adjunct professor in the School of Food Science and Environmental Health at TU Dublin. Dr. Maria is a current coordinator and PI of several EU projects, including Sea Solutions. She works on developing new products with health benefits through identification and characterization of bioactive ingredients from different marine and terrestrial resources. To do this, she uses a combination of different bioassays that she established at Tigarsk, including those relevant to maintenance of heart health, prevention of type 2 diabetes and mental health issues, prevention of atherosclerosis, inflammation, and pain. Dr. Maria has also developed a method to screen methane mitigating abilities of algae in vitro. Today, she will share some of her research in algal proteins and bioactive peptides. The floor is yours, Dr. Maria. Thank you.
Thank you very much, Dina, for that very nice introduction. I know from the practice session yesterday that there may be some issues with the format. I think Arul has put it up. I don't know, Arul, if you can move to the first slide, please.
I just shared your screen now. I will just see whether it is moving or not. 1 minute. Yeah. So
yeah,
perfect.
If you can just go to the first slide, please. Yeah, great. Thank you. So as Dina said, I'm a senior researcher. First of all, I'm delighted to be invited today to present, and hello to everyone in DCU and the University of Mauritius and to everyone online. Hopefully some good collaborations can come from today. So I was invited to speak on the topic of algal proteins and bioactive peptides. Just a little bit first about our research centre. If you could move to the next slide, please. Yeah, TAGISC is the Irish Agriculture and Food Development Authority of Ireland. It's the national body that provides integrated research and training advisory services to our agricultural community and food industry and rural communities. We have about 1,100 staff at 55 locations nationally and an operating budget of about 160 million. I'm located in Teagasc Ashdown, which is here in Dublin. And we have several other centres in Cork, in Meath, in Athenry in the west, and we have several advisory offices around the country. Next slide, please. So in terms of marine research, I suppose it's underpinned by a number of nationally and EU funded projects that we've received over the years. The first of which was Nutrimara, which was an all-Ireland initiative involving all of the universities, which TAGS led. And it was all about determining the potential of marine ingredients, including algae, as functional foods. Following on from that, we've had other projects like U-Protein, Algae for IBD, both of which look at algal ingredients as sources of nutrition and for health benefits. And we also have, as Dina mentioned, the Sea Solutions project, which I coordinated, which concerned the use of algal ingredients as potential feed additives to reduce methane emissions. So our research really is underpinned by the skill set highlighted there in the centre of your screen. And it's underpinned by blue growth and sustainability where possible. And we also have an interest in feed and pet feeds for companion animals. Next slide, please. Yeah, so as you probably know, if you're based in Europe, the European Commission supports blue growth initiatives and encourages a farm-to-fork approach. And because of this, there has been active promotion of algal as a source of food, in particular protein, over the last number of years. Next slide, please. Algae include seaweeds naturally, and here in globally, we have a huge resource of seaweeds, highlighted by the red spots on the map of the world here. And in Ireland, the interior map actually shows the real size of Ireland. So if you include our blue zone, our marine area, it's about 3 times larger than our land mass. And along the west coast of Ireland, we have about 600 different species of algae. And these include things like dulse, carrageen moss, and kelps. Next slide, please. In terms of what we do with these seaweeds, traditionally there's a focus on harvesting. And as you can see from the table here, we are third, I guess, in Europe in terms of the tonnage of species that we harvest. And these species include things like rockweed, European kelp, and red seaweeds primarily. And of the kelp, I suppose Ascophyllum nodosum will be the seaweed that's most largely harvested. And in terms of red seaweeds, then we harvest around 100 tons per annum of Chondrus crispus, marl, and Palmaria palmata. Next slide, please. In terms of cultivation, we lag behind some of our European and definitely our international colleagues in this area. So the species that we focus on really in Ireland include Laminaria digitata, Alaria esculenta, and Saccharina latissima. And these seaweeds may not really be the most valuable for use as foods. So they're traditionally used really as animal feed, and they're just usually harvested, ground, and sold as feed additives. There is an issue with some of them in terms of heavy metal content, which can prevent their use as animal feeds also. Next slide, please. Yeah, so to get to protein, I suppose, the different seaweeds have different protein contents, and the reds are known to have the most protein. They have been reported in the literature, and indeed we found from our research here that they can contain varying amounts of protein and up to 47% protein on a dry weight basis. Green seaweeds also contain a significant amount of protein. They can contain up to 26% protein, and the brown seaweeds have a lesser amount of protein. Now, these seaweeds basically compare favorably with dairy products, for example, which would be consumed normally for their protein content. Milk, for example, per glass contains about 4% protein. So seaweeds contain a lot more protein than milk. And many of the seaweeds I've listed here as red, green, or brown, they often contain all the essential amino acids and some acidic amino acids, and indeed amino acids with additional health benefits. Next slide, please. Yeah, so also from market research studies, seaweed protein is quite a hot topic now, and it's estimated that the market for seaweed proteins will increase by about 11.8% by 2030. And red seaweeds and food-derived edible seaweeds, so foods that are approved for consumption by EFSA or whomever globally, are set to drive this market. Next slide, please. So that all sounds fantastic, but there are caveats, of course, associated with all of this. So seaweeds, as most people online will know, have a very resilient and strong cell wall composed of different polysaccharides. So some of the earlier speakers mentioned the use of these polysaccharides for packaging and other applications. But in terms of protein, they can add benefits as prebiotics, for example, in a food formulation, but they hinder the extraction of protein using methods that are traditional, okay? And consumers in general are less inclined to eat seaweeds whole as a source of protein. So we would like to extract the proteins from the seaweeds because it makes them more user-friendly and they have more applications when we can extract the protein. It's a more consumer-friendly product for applications. But this is not an easy thing to do, but we are trying. And if you could move to the next slide, please, I can maybe discuss some of the strategies that we use to try and extract the proteins from different seaweeds. So we use both, and we have used and published on these, we use both physical and chemical treatment strategies to extract proteins from different seaweeds here in Teagasc. So we have used what we call the traditional protein extraction method, which involves sonication of the biomass and then precipitation of the protein using ammonium sulfate. We've also used high pressure processing, which helps to break the cell wall and then the supernatant should contain more protein. We've used autoclave to basically heat up the cell wall and release the protein. And we've used technologies like microwave assisted extraction, electroporation technologies or PEF, and enzyme extraction via hydrolysis. Next slide, please. So I'm going to talk more about hydrolysis today because, to be honest, it's one of the technologies that we've seen the best results with, and it's a very scalable technology if you know what you're doing. So precision hydrolysis can help to overcome sensory issues with seaweeds, for example, if you can extract out the protein. And it can also increase the incorporation rate of dietary amino acids into the diet, which can improve skeletal muscle protein. When you hydrolyze a protein, be they from seaweeds or other sources, it can also increase the technofunctional. It can also sometimes reduce the technofunctional attributes, and it can increase the bioactivities associated with that protein. Okay, so hydrolysis can generate bioactive peptides. For those people who don't know what bioactive peptides are, they're sequences of amino acids between 2 and 30 in length that can be released from any protein source using enzymes and hydrolysis or high pressure processing or chemical hydrolysis, okay? And they impart a health benefit to the consumer that goes above and beyond basic human nutrition. Next slide, please. Yeah, so bioactive peptides can be found in the body naturally because you have your own endogenous proteins and you have enzymes in the body. So they're endogenous proteins, but food-derived proteins or seaweed-derived proteins are basically exoproteins, and they're basically consumed in the diet, and they have to be released from the parent protein to impart the health benefit. So that's what we do when we use hydrolysis. We basically break down the protein into peptides, and some of these peptides then have bioactivities. Next slide, please. Yeah, so as I said already, any protein source can be a source of bioactive peptides, and hydrolysis is a methodology used to generate them. From seaweeds and other protein sources like dairy would be the most famous resource for bioactive peptides. Next slide, please. Yeah, so food-derived bioactive peptides, as I said already, they can be found in things like dairy, and they're found in the food and have to be generated using hydrolysis and other methodologies. They can be released by gastrointestinal digestion, but in general, they're not present in sufficient quantities to impart a health benefit to the consumer just when you consume a whole protein. And they have a myriad of health benefits, including things like antioxidant activity, antimicrobial activity, heart health benefits through the action and inhibition of enzymes such as ACE1, renin inhibitory, and opioid activities. Next slide, please. Yeah, so 2 health problems that are huge at the moment in Europe and in Ireland include type 2 diabetes and hypertension. So type 2 diabetes, there are about 60 million people with diabetes in Europe. 10.3% of men and 9.6% of women aged over 25 are affected and can develop type 2 diabetes. And in general, about 3.4 million people die annually of diabetes. So this will be a target for our bioactive peptides, and I'll speak a little bit more about this in a minute. Next slide, please. So hypertension, as I said, is also a huge problem still, even though we're maybe down the list now of countries in Europe that are affected by high blood pressure. Croatia has the highest incidence of high blood pressure in Europe, but it still affects 22% of people over the age of 15 in the EU. And there are about 10.4 million deaths from hypertension or high blood pressure worldwide annually. And dietary approaches to stop hypertension, or DASH, is a key strategy in Europe to help improve our health. Next slide, please. Yeah, so how does seaweeds and bioactive peptides relate to prevention of hypertension and prevention of type 2 diabetes? So the peptides that are derived using hydrolysis methods from seaweed proteins can prevent the action of enzymes within systems of the human body. The systems of the human body I'm speaking about are, firstly, the renin, angiotensin, aldosterone system. So this system controls saltwater balance and blood pressure in humans and many mammals, okay? The two enzymes key in this system to control of hypertension are renin and angiotensin converting enzyme 1, or ACE1. So our bioactive peptides, if they have ACE1 inhibitory or renin inhibitory activity, inhibit the action of renin, which is the rate-limiting step in the formation of hypertension in the body, or they can limit the action of ACE1, which converts angiotensin 1, a vasodilator, or something that dilates your blood vessels and maintains normal tension in humans. It prevents the action of ACE on angiotensin 1, preventing the conversion of this vasodilator into the vasoconstrictor angiotensin 2. Okay, so a vasoconstrictor obviously constricts your blood vessels and it's harder for blood to pump through your vessels, so therefore blood pressure increases. In terms of type 2 diabetes prevention, bioactive peptides derived from seaweeds and other sources can have dipeptidyl peptidase 4 inhibitory activity. So they can inhibit the action of this enzyme, which basically can convert the gastric inhibitory peptide GIP or the glucagon, I can never say that properly, glucagon peptide like 1 or GLP-1 into, it can inhibit these and therefore prevent the release of and the stimulation of insulin secretion basically into the blood. So our bioactive peptides from seaweeds, we want to target these three enzymes, I suppose, as a strategy to improve health through nutrition. Next slide, please. So yeah, we first started work on this whole area many years ago now, back in 2008, 2009, and a PhD student that worked with me here, Kieran Fitzgerald, looked at the red seaweed Palmaria palmata for this purpose. So as part of his PhD, Kieran took this red seaweed and generated different protein hydrolysis using different hydrolytic, proteolytic enzymes, and the traditional ammonium sulfate salting out method to generate protein extracts, which were hydrolysis essentially containing bioactive peptides. And he then assessed these hydrolysis and the identified peptides for renin and ACE1 inhibitory activities. And this work was carried out in conjunction with University College London, the School of Pharmacy in the UK. Next slide, please. Yeah, so as part of this work, we identified a number of different renin and ACE1 inhibitory peptides. These peptides were assessed in an in vivo model for hypertension, the spontaneous hypertensive rat. And when fed to the rats, the hydrolysis containing the peptides, the active peptide itself, a tridecapeptide, they both had a significant hypertensive effect in spontaneously hypertensive rats compared to the controls. Captopril, a known ACE1 inhibitory drug, which is known to reduce hypertension and is prescribed regularly for people for hypertension production, and compared to saline. Okay, so this was a very good result for us. Next slide, please. Yeah, so we obviously wanted to put this protein hydrolysis ingredient with hypertensive effect into some kind of food carrier. And at the time we selected bread as a food carrier. So why bread? I guess bread is widely consumed globally and it's a staple in most people's diets. So different bread types were manufactured, including buckwheat and just traditional white flour. And then we assessed the ability of the bread to reduce hypertension, and it also resulted in a reduction in hypertension when consumed. And in terms of the sensory acceptability of the bread, it was deemed relatively acceptable by our consumer panels. Next slide, please. Yeah, so basically this was the rationale for selection of the bread as a food carrier for hydrolysis. And additionally, wheat protein, which most breads in Europe are made from, can be deficient in essential amino acids such as lysine. So the addition of the hydrolysis from Palmaria palmata could increase the lysine content of the bread and make it additionally not just antihypertensive bread, but a nutritionally beneficial bread as well, providing all the essential amino acids to the consumer. Next slide, please. Yeah, so we also did some work with Chalmers University in Sweden, using our different extraction methodologies, traditional protein extraction with ammonium sulfate, precipitation of the protein, accelerated solvent extraction or pressurized liquid extraction, and the pH shift process, which basically employs HCl and NaOH to increase and decrease the pH of a solution containing your seaweed to burst the cell walls of the seaweeds and release the protein. And this work was also published by Hannah Harrison, who was a PhD student at the time in Chalmers. Next slide, please. And if interested, you can follow up on it there. But to summarize the findings from this result, we found that when we applied the pH shift method to Perfora species, which is known to have a high content of protein, we got the greatest yield back of protein. But when we applied the same method to Ulva, we found that the traditional method using ammonium sulfate precipitation yielded the greatest amount of protein. An advantage of the pH shift method was that it concentrated the fatty acid content in the protein extract, and this could be beneficial for some health applications as well. Next slide, please. Yeah, so in my work here to date, we still use hydrolysis a lot. We customize different hydrolysis methods to apply to different seaweeds to enhance protein extraction. And then we characterize the resultant bioactive peptides. So we use mass spec in conjunction with partners in CSIC in Spain to identify peptides within our hydrolysates. And when we do this, once we know the peptide sequences, we basically can determine, using in silico methods, if there is any potential bioactivity associated with those peptide sequences. So we do this using the schematic I've outlined in the following two slides. We use like the FASTA program, and we are UniProt or PubMed, and we access the FASTA file of a certain protein if it's available. From there, then we would try and break up the protein using in silico hydrolysis using XPC peptide cutter. And then we would assess the novelty of the resulting peptides by consulting databases like BioPep, which is point 3 here. Next slide, please. We can then predict the likelihood of a bioactive peptide derived from a protein source as being bioactive using programs like Peptide Ranker. And then we can also identify if they have potential hypotensive effects by monitoring the amino acid composition of the peptide and the location of the bulky side chain aromatic amino acids within the chain of the peptide sequence. We can predict the digestibility and bioavailability of peptides using in silico digestion methods, again using XPC peptide cutter. And we can even predict the flavor potential or umami imparting potential of peptides using different programs. Next slide, please. So it's a very useful strategy for identifying bioactive health benefits of protein hydrolysis, be they from seaweeds or from other sources. So an example of where we've used this in relation to algae relates to recent work we did concerning the anti-hypertensive activity of Perfora hydrolysate that we developed. So we used in silico analysis and we predicted the peptides found in a hydrolysate. And then we actually confirmed the bioactivity of that hydrolysate in both in vitro ACE1 and renin inhibitory assays, and also in vivo spontaneously hypertensive rat studies. And again, we found that the hydrolysate had a hypotensive effect comparable to captopril and better, obviously, than the negative controls. Next slide, please. Yeah, so we've published a number of papers in this area, not just for hypotension prediction, but also for anti-inflammatory predictions and pain predictions. Next slide, please. Yeah, we've also extracted proteins from microalgae using hydrolysis with different enzymes. And here I just highlight viscozyme and alkalase as two that we've used to basically determine, to make an ingredient with technofunctional benefits. Next slide, please. Next slide, please. So that's all well and good, but I suppose essential to any protein ingredient really is the amino acid content of the protein. So the first thing we always do is look at the amino acid content of our different seaweed protein ingredients. And that's important nutritionally, not just for the territory benefits of the protein, but nutritionally because it predicts the quality of protein in terms of nutrition. Next slide, please. Yeah, so like for food companies, for example, if they're making a nutritional protein claim like contains 10 grams of protein per serving or high in protein, in the US or in Europe, EFSA in Europe and the FDA in the US, they basically suggest and actually demand that you have to provide the percentage daily value for protein in the nutrition label. And that percentage daily value basically has to be based on the corrected protein value and not the amount that is declared in the nutrition facts label. Okay, so to do this, we have to predict the percentage daily value protein content of a hydrolysis. We have to use FAO recommended methodologies, and one of those FAO recommended methodologies was the protein digestibility corrected amino acid score method. Okay, and this basically predicts the protein content of a product, and it's calculated based on the protein of products made for adults and children of 1 or more years of age, and you have to multiply the declared amount of protein by the PTCAS value. So recently the PTCAS value was replaced by the DS value, which I'll explain in a moment, but basically you should know the PTCAS value for your proteins. Next slide, please. So what the PTCAS basically is the protein digestibility corrected amino acid score, and it basically considers both the indispensable amino acid content of the test protein and its digestibility. So it basically predicts possibly the bioavailability of the protein. Okay, the amino acid score is equal to the content of the first limiting amino acid in your protein, and it has the corresponding content of this amino acid is then compared to WHO, FAO, and UNU reference patterns for protein requirements for preschool children. So PTCAS is basically the protein digestibility multiplied by the amino acid score, and that's really a measure of something that is a good source of protein. Okay, and it enables a company to make these kind of claims if you have the protein digestibility corrected amino acid score calculated for your protein. Next slide, please. So yeah, in terms of monitoring it, there is or assessing PTCAS, there are both in vivo and in vitro methods, and I've just highlighted the steps here for both of those. Next slide, please. So in vivo, the protein digestibility corrected amino acid score involves the use of rats or pigs, and these measure the true digestibility of the protein. And then if you know the corrected amino acid score, you can calculate the PTCAS value. Next slide, please. But there are also in vitro methods that you can predict the PTCAS value of proteins, and it's an Irish company, Megazyme, produce a kit that can be used to generate PTCAS values for any protein source. Okay, and it's basically a digestion step that predicts the digestibility of your protein, and then you calculate your amino acid content of the protein as well, and therefore you can calculate your PTCAS value. And there is some evidence now. In vitro methods vary for monitoring PTCAS values. This is a commercially available method, but there is a large group in Europe here known as Infogest that are researching alternative to animal models for predicting PTCAS and DS models. Next slide, please. So yeah, in terms of seaweed, I guess there is very little known about the PTCAS value or indeed the DS values of seaweeds or seaweed protein extracts. Here I give an example of a few seaweeds where the PTCAS values have been calculated. And if you look at the second last column, you'll see that most of the values are below 0.7. So to be considered a good protein source, the score in terms of PDCAS should be close to 1. So dairy and meat proteins, for example, or fish proteins, would have values of 1 or 0.99 or 0.92. Soy also has a very high PDCAS value. So to be considered a source of good protein, the value should be higher than indicated here for seaweeds. Okay, so it's likely that if you were to use any of these seaweeds, and these are whole seaweeds, and state that they're a good source of protein, you would have to make blends with other ingredients to do so. Next slide, please. Yeah, so the in vitro and in vivo methods, there is good comparison for some protein sources, but there is ongoing work in this area, as I said, to develop new alternatives to the animal models for measuring PDCAS and TS values. But for chickpea proteins, for example, there is correlation. Next slide, please. I'm conscious of time. Yeah, so recently the PDCAS method recommended by FAO and WHO has been replaced, I suppose, or it's recommended that it is replaced by the digestible indispensable amino acid score method. And this basically is a protein quality method proposed in March 2013 by the Food FAO to replace the current protein ranking standard, the PDCAS. Okay, next slide, please. So basically, the ratio of digestible amino acid content in the food to the same reference pattern taken from each specific amino acid requirements is determined. And then the lowest value across amino acids is multiplied by 100 to convert the ratio to a percentage. And this percentage represents the DS value of the food. Samples are taken from in vivo digestibility assessments from the ileum, and it's usually a pig model that's used to calculate DS, okay? And to make claims on proteins, like for example, if your DS value is less than 75%, you can't really make a protein quality claim. If your DS value is between 75 and 99%, you can make claims like good protein quality. And the score of 100% or more suggests that it's an excellent source or high protein quality food. Next slide, please.
Dr. Maria,
yeah, nearly finished now. This slide, sorry, Dina. So yeah, and then just I'd like to refer you to this recent publication by the FAO and the International Atomic Energy Agency. It's basically the technical report from a meeting that was held in Vienna in 2022, in which I was a participant, and it makes recommendations about the way forwards for developing a protein database and protein requirements for individuals. So obviously, if seaweed is going to be a future protein resource, it will have to comply with the recommendations of FAO and WHO in terms of the nutritional requirements laid out in these documents. So I'd like to thank everyone for their attention. Sorry if I went a little over time, and I'm happy to take questions.
Thank you. Thank you very much, Dr. Maria, for this presentation. It was very interesting.